Okay, great. Great. Okay, so thank you very much, Pradeep, for the kind words, and it's for the invitation to speak for you today. It's really, really nice to be here, and it's nice to see some familiar faces in the attendance list. And you know, it's I hope to just give you an overview of some of the work that we've been doing, and yeah, give you a flavour for what we've been doing. So really, we I like to start a talk like this with this slide. I mean, this really summarizes in a certain way, you know, what certainly I'm you know, the goals of my research, what I see as the things that are important, you know, developing innovative machine learning methodology, applying it to big data, to neuroscience, really aiming to have impact on problems that really matter. So, you know, that's really a mission statement, if you like. I'm going to talk to you about three things today. I'm going to talk to you about using biomarkers for stratifying cohorts, modeling variation using in big data cohorts using normative modeling, and some approaches for methods development. I should say that all of the work today has been put together by a fantastic group of people, and I'll try to give due credit where due credit is due. And also, just to note that these things interact, these themes really run through all of the work that we do at the Donders Institute. So the notion of biomarkers has a very long history in psychiatry. Basically, the notion is that we want to use biological measures to make a prediction with regard to diagnosis or treatment allocation at the level of each individual participant, much in the way that blood tests, for example, are used in many other areas of medicine. This departs a little bit from the classical scientific inference dogma, which aims fundamentally to provide group level inferences, so differences, for example, between patients and controls in, for example, a GWAS study or statistical parametric mapping. The problem with this kind of approach, while it's very good for discovery of mechanisms, from a clinical management point of view, the problem is that clinicians need to treat individuals, not groups. So a number of years ago, we started working, and others as well, started working on using pattern recognition methods to discriminate clinical cohorts. So, for example, patients and controls. And this, the central idea is that we aim to learn some kind of decision function in some multidimensional space that enables us to separate clinical cohorts as accurately as possible, such that we can then make a prediction as to which diagnostic label, for example, a new participant belongs to. And of course, the central idea is that we've moved from making inferences about groups to making inferences about individuals. And there are also we can also use this this kind of methodology for quantifying group separation. Hold on one second. Oh, sorry, my kid's just trying to ask me a question. We can also use this kind of approach to quantify separation between clinical groups in a multidimensional space. This is probably the first study that I worked on, and I'd just like to sort of use this to illustrate that this idea has been around for a very long time. In this study, we used fMRI data derived from patients with depression and a support vector machine classifier to discriminate, first of all, patients from controls, and also treatment response. And even though this study has, you know, an embarrassingly small sample size, it was, to my knowledge, the first machine learning study that was published in a major psychiatry journal. Since that time, there have been hundreds of studies that have used this kind of pattern recognition approach, and I would refer you to some of these review papers. That was one, for example, written by Thomas Wolfers, who was a PhD student in our group. Basically, this kind of pattern recognition approach has been applied in hundreds of different contexts, virtually any brain disorder that you can consider, and predicting many different kinds of clinical variables. Hold on one second, please. And so this kind of approach has been used, as I said, in many different contexts. Now, when Thomas reviewed this literature, he found something surprising, and I'm plotting here the accuracy that across different studies with the number of patients in the x-axis and the accuracy in the y-axis. And you can see some quite disturbing trends. So first of all, you see there's a cluster of small studies on the left-hand side. And as you increase the sample size, the accuracy decreases, which of course, from a machine learning perspective, is very counterintuitive. You would expect the accuracy to increase as sample size increases. And really one of the reasons for this is heterogeneity within the cohorts that we're studying. And as the sample size gets larger, we sample more and more of these different heterogeneous phenotypes. And it's not difficult to think of where this might come from. So for example, on the one hand, we may have clinical comorbidity, participants with more than one disorder. There's variation across the presentation of different individuals with the same disorder, and there's also heterogeneity at the level of mechanisms. So addressing these at this heterogeneity at these multiple different levels is I think, a major challenge for the field at the moment, which motivates the next piece of work that I'm going to talk to you about how can we kind of address this. And this is of course not it's relatively well understood that these this problem is not unknown, and is really at the heart of some of these really large international initiatives, for example the ARDOC initiative, that aim to develop for research purposes new ways of classifying disorders based on neurobiology and behavior. And this all sounds good, but I'd like to sort of pull this apart a little bit. And on the one hand, ARDOC has provided us with a very precise way to break down cognitive systems across different levels of analysis. It's a very detailed description of psychopathology. But the clustering notion is not really new at all. People have been trying to do that for decades, actually. And we reviewed the literature a number of years ago. And hold on one second, please.
I guess quarantine life, right? Yes.
I apologize for that. So basically, the point is that people have been approaching this problem for decades, and we haven't really been able to make headway. And to illustrate, I'd like to take one particular example, which, and I think, sorry, one of the reasons for that is that frameworks such as ARDOC don't really specify how we move from these heterogeneous symptom groups using biological measures to somehow produce clusters that more closely relate to underlying biology. So that's not specified by any of these initiatives. To illustrate that, I'm going to take one relatively high-profile study that was published in Nature Medicine a few years ago that aimed to use resting state functional connectivity to derive putative blood biotypes. So the authors of the study used canonical correlation analysis to derive mappings between symptomatology and resting state functional connectivity using canonical correlation analysis. And you can see we have very strong correlations between these two domains, at least ostensibly. And these biotypes, which you can see at the bottom here, were then applied to predict treatment response in terms of TMS, and they're also present in a cross-diagnostic setting. Now, Richard Dinger, who won the Replication Award, is a member of my group who won the Replication Award in 2019 for his work in replicating this study. Started off from a very sort of low-tech perspective, if you like. And so these are the biotypes that were produced in the Drysdale study. And one of the things that is a bit hard to see from this figure is that there are some of these dots that are grayed out. And when you do something like as simple as coloring all the dots the same color, you can see that the evidence for distinct clusters is somewhat more equivocal than the study would suggest. Richard went on to replicate this, at least from the methodological perspective, this study using the NESDA sample. I'll describe that in a minute. We also think that there were some problems with the statistical analysis of this paper. There were lots of in-sample tests. They didn't test the whole analytic pipeline and so on. So Richard developed a more, I would say, rigorous statistical evaluation of this pipeline and applied it to this sample. So the NESDA sample is a large cohort study in the Netherlands, which required many different kinds of measures and followed a number of patients up longitudinally, which is, I would say, makes it a pretty good candidate for a replication of this particular study. Now, the first thing that Richard found when he applied this pipeline is we find again the same ostensibly strong correlations derived in sample. But one of the things that Richard showed is that there's nothing particularly surprising about that. So when you do a permutation test, you can see that the null distribution is very sharply peaked around about 99, a canonical correlation of about 99, and yet out of sample, it was the canonical correlation drops to about 0. The loadings that we use to derive the clusters are also very unstable. So this is showing small perturbations of the data lead to basically totally different representations, and this of course propagates through to the clusters. So when we have while a clustering solution can be derived in sample, when you apply it to a new data set, it's effectively it doesn't survive that. And similarly, Richard also did lots of evaluations in terms of the separability of the clusters, and even though the usual cluster order, the bottle order statistics for determining the number of clusters are relative in the sense that they test 2 against 3 and so on. It's pretty uncommon to test against the null hypothesis that there are in fact clusters in the beta at all. So at least in our hands, this kind of approach, which at least in our data, it would suggest that the evidence for any kind of biotypes is relatively equivocal. It emphasizes the need for validation and also this idea about testing against this kind of null hypothesis. It should be said, however, that can be quite hard and it might not even be possible in certain circumstances. Following on from this, we had a long discussion with the authors about this kind of approach. They said, well, it's different data and so on, and quite helpfully, the authors of this paper effectively did the same analysis that we recommended, or that Richard recommended, and showed the same thing. So you can see here, as these are in-sample canonical correlations, and as you increase the number of features, then the out-of-sample, which is the green distributions, sinks to 0, even if the number of features is about the same size or just slightly smaller than the number of the sample size. The thing to take from this, I mean, this is a really interesting discussion. I think in the end, quite a constructive discussion about the use of canonical correlation. I mean, it's very prone to overfitting, so it needs to be used with caution. And I would recommend anyone sort of interested in this kind of approach to take a look at this dialogue, because I think it contributes quite something to the literature. So the last, so that sort of leads us on to this notion about how can we move forward. So I've been talking so far about these two different approaches, sort of this case control approach that we might use a classifier for, or some kind of clustering approach. And of course, that's only one possibility. There's many other things that could be happening in the data. So I think it's reasonable to assume that the notion of distinct case and control populations is relatively unrealistic. But this has kind of been the clustering approach has been the go-to method in response to that. But it's important to recognize that that's not the only possibility. We may have other scenarios as well. So certainly in psychiatry, we may have something more akin to a spectrum of functioning, or we may have a scenario where case population is extremely heterogeneous and through a combination of different mechanisms, different presentations, and so forth. And this is one of my favorite figures. This is from a genetic study, and this kind of shows a conceptual space representing different disorders that I think quite accurately reflects, what is probably happening. Lots of overlap between different disorders, very fuzzy boundaries, and so forth. So our approach, one of the approaches that we've kind of invested in recent years to try and address this problem, generalizes the notion of pediatric growth charts. So if I'm sure most of you will have seen this kind of
Oh, it's maybe the, oh, he's back. Okay.
Yeah, sorry about that. I dropped out. My connection dropped.
It's okay.
Hold on one sec. Okay. I'll just get my slides back. Hold on one sec. Okay, here we go. Right. Okay, so we're talking about normative modeling. Can you see my slides? Yes. Yeah. So the idea is that if we want to learn about child development, what we can do is we can map individual development as against centiles of variation in the population. And the idea would be that one might become concerned if development drops into the lower centiles or equally into the upper centiles. And the idea behind normative modeling is that we substitute these variables for things that might be of interest in psychiatry. So, for example, to estimate mappings between biology and behavior, and we quantify individual variation in much the same way. So in a little bit more detail, what we aim to do is we aim to estimate some kind of mapping between an arbitrary set of predictive variables and biology in using some kind of probabilistic regression method that provides us with an estimate of uncertainty and an error for each participant, so that we can then use those statistical measures to derive subject level statistics as to how each individual participant deviates from the expected pattern. Now, if we do this across a number of different mappings, describing, for example, different brain systems, different cognitive variables, etc., what we can do is we can build up some kind of normative space that provides a reference distribution for a population of our choice, and we can get a deviation in each of these measures for each of the mappings that we estimate. We can derive some kind of atypicality score using, there's a number of different methods to do that, but one that we use is using extreme value statistics, so that we can then detect disease fingerprints. And the point is that while we may use this as a pre-processing approach for clustering, it doesn't necessarily assume that clusters exist in the data. I'm going to illustrate that with an application by Mariam Zabihu, who is on the call, as well as Richard is. In this application, Mariam was interested in parsing the heterogeneity that underlies autism in terms of biology. And it's a very good candidate for this kind of approach because it's very heterogeneous, has lots of different genes that have been implicated, and also different brain systems. She used a very large sample for that. This is a European consortium that aims to really quantify variation across the whole spectrum, from low functioning to high functioning. And so what Mariam did is first, we'll talk in terms, so Mariam's analysis focused on cortical thickness. And I'll show you first of all what we see in the data when we take a classical case control approach, and the answer is not much. So there are virtually no differences despite good statistical power for detecting group differences. We don't see many differences, and for a 50 million euro project, that was quite a disappointment, to say the least. So what Mariam did is she estimated a neurodevelopmental normative model that quantifies variation across each cortical location as a function of age and gender, plus some other sort of nuisance covariates, if you like. And she estimated this model, which quite nicely recapitulates the brain development literature. So roughly speaking, the cortex thins as we mature in a relatively monotonic fashion. It becomes interesting when we plot the participants with autism against this reference model. And what Mariam found is that in contrast to the case control findings, there are many differences. In fact, virtually the whole cortex has differences in at least some individuals. But if you look at the scale at the bottom, you'll see that it's very idiosyncratic in the sense that the same deviations are not reported in the same individuals, which is one reason why you don't see many differences. And I'll show you another reason in a moment. So what Mariam did is she took these images and then fed them into a spectral clustering algorithm, which is quite well suited to high dimensional clustering problems. And she derived a set of clusters. Now I'm putting biotypes in inverted commas here because I don't think we've really demonstrated that the clusters are distinct or that they're really exist in nature, but they're quite a useful mechanism to understand the heterogeneity that's in these that we see in the data. So please bear that in mind. When Mariam looked at the profiles of these different clusters, we see quite a striking pattern emerging. So you can see that of these 5 clusters, 3 have reduced cortical thickness and 2 have increased cortical thickness. And this is all in participants with the same clinical label. And there's also regional differences and so on between the clusters. And that really explains quite nicely why we don't see any case control effects, because we're mixing all these different biological signals in the same cohort. Mariam took this further and then related these to these biotypes to both symptoms and genetics. And you can see at the top we have some modest associations across the whole cohort. Yet those associations with these are different symptom domains in the different bars. It's different autism symptoms and also different comorbidities. And it's evident that as we break these down across the clusters, these associations always get stronger and we see more significant associations, which suggests that the associations with symptomatology and biology are only present in a subset of individuals. Just to note that we've done this in lots of different disorders, in ADHD, schizophrenia. I don't have time to talk about them all, but there's a similar theme emerges throughout all of these different applications. So for the last part of this talk, I'd like to talk about approaches that we've done. A big focus of our group is on methods development, and I'd like to talk about that a little bit to facilitate some of these objectives that I've outlined so far. So let's start with normative modeling. So normative modeling, the way I've been describing it so far, is based on using Gaussian process regression, which I'll just walk you through this. So we have some kind of function, and we assume Gaussian residuals, and we have a Gaussian process, which you can write in this way. It turns out to be a Gaussian distribution over some kind of mean function and a covariance matrix. If you know anything about Gaussian distributions, then you'll know that in order to make predictions in this context, it's necessary to make a conditional Gaussian distribution for which it's necessary to invert this matrix, which, as the sample size increases beyond a few thousand, becomes pretty problematic. It doesn't work both in terms of memory and in terms of time. So what one of the members of my group, PhD student Charlotte Fraser, has been working on is developing a way to get around this. So she started off with replacing this Gaussian process with a linear basis function model, for example, using a B-spline basis. This is not particularly new. There's lots of people that have been doing this, but it's much more scalable. She combines that with using some kind of warped likelihood, and that allows us to model non-Gaussian distributions. So the difference between this linear basis function model, where we have basis expansion, I'm denoting that by phi, and this one is that we also warp the target variables. And so that enables us to learn some kind of nonlinear monotonic mapping between some non-Gaussian signal and pass it through this function, and we end up with something that can be modeled in a Gaussian fashion using all the convenient machinery that we have for that in machine learning. This is by construction specified in a way that the function is invertible, so we can move in both directions in a closed form way, and we can also compose these functions together, which gives us a very expressive machinery for modeling very complex distributions, at least in practice. So Charlotte applied this to the UK Biobank data. The first thing is that we have excellent computational scaling relative to the Gaussian process model. So you can see that this is the computation time as a function of the sample size, and you can see that it's almost constant relative to the very poor computational scaling of the Gaussian process model. I'll show you an example of, just in the interest of time, about this kind of normative modeling approach for one phenotype that's derived from UK Biobank. This is for a particular phenotype that describes the white matter hyperintensities. It's lesions in white matter, basically. You can see that this phenotype has a very skewed, very non-Gaussian distribution, and the fit of the Gaussian model is obviously very bad. When we apply this likelihood warping technique, we end up getting a normative model that much better reflects variation in the population, captures the different nonlinearity, heteroscedasticity, and skewness of the distribution. And then we can take advantage of the longitudinal data that is provided in UK Biobank to get a feeling for the development of these different trajectories, how people age over time. And what I'm showing here is I have colored all the subjects so that each of these lines is a person. So that's the white matter and hyperintensity load at time point 1 and time point 2, which is a few years later. And I'm coloring all the people that have a deviation larger than half the standard deviation with respect to what would be predicted by the normative model. And it turns out that these atypical individuals have a much more severe cognitive deterioration than people. So this maps onto cognition, basically, and it does so in a way that is is cleaner than just taking each time point individually in a cross-sectional fashion. Another challenge, this is work from Mozikia, is how do we model site effects. I mean, it's quite clear from this figure that there's very severe site effects present in most neuroimaging derived phenotypes. And it's quite clear also that if we were to fit a normative model in this kind of data without accounting for side effects, we'd have centiles that are hopelessly wide. So the challenge in normative modeling is not just modeling the mean, it's modeling the variance as well, as I've shown you. So we have to do something about that. The conventional approach is data harmonization, which is where we say we take data from different sites, we try and match them up using some statistical model to do that, and then we end up with data with side effects removed. And the classical way to do that, of course, is combat. The idea behind combat is we adjust the data in this form. There's a parameter that describes an additive site effect, a multiplicative site effect, and then the signal of interest is captured in this way. And I'll explain how it works in the next slide. If you want to adapt combat to new sites, you need to update the parameters. So you need to have access to the data, at least in the most classical way that this is used. What I can say about it is that it works pretty well if it's used in the correct way. So you say that this is a large multi-site sample. You can see this is again cortical thickness. You can see site effects are largely removed after applying combat. There's also different nonlinear versions. This is one from Christos Stavitsikos' lab. And one thing I'd like to point out is that there are strong correlations between in basically every real neuroimaging sample that you're likely to encounter between age and the thing that we're basically between age and size. So it's important to deal with that. I would like to pause for a second here and just recommend to anyone that hasn't read this paper. This is really a classic paper. It's like 20 years old, but it's really describes some important caveats about deconfounding, regressing, dummy variable regression, and so on. And if, for example, you've ever been asked to regress out IQ between clinical groups that, for example, patients and controls, this paper tells you very clearly why that's not always a good idea. And I'd also recommend to take a look at Richard's talk later on about different deconfounding methods in predictive models in general. So back to combat. I'd like to sort of just break down what combat actually does and show you in some circumstances there are theoretical problems with this harmonization approach. And I stick with a linear context, but just bear in mind that this translates to nonlinear and deep learning based methods as well. So if we have the variance that we're trying to model, and I'm going to break that down into three components. So we have the known variance. That is things like age. They're things we can put in the design matrix and try to predict. There is unknown variance, which might be clinically interesting, but we don't measure. So for example, subtype, symptom severity, etc. And then there's site effects, and those are the things that we want to remove. It's really important to recognize, as I pointed out previously, that these things are potentially correlated. So, we need to deal with that in some way. So what combat does, in simple terms, it's not done in a sequential fashion the way that I'm explaining it, but this is just an outline of what it does. Basically, first we remove the known variance. So we remove this part here. This is what this part of the equation does. We remove the batch effects, and then we put the known variance back in, or the experimentally relevant variance back in. You can see that this approach is vitally dependent on the correlations between different things that we're trying to measure. And the way that I've drawn it is that we lose the part of the variance that correlates with the batch effects, and we also remove this part of the variance. We remove this part of the variance when we shouldn't. So combat has a difficult time dealing with these kinds of different correlations, because we have to basically tell it in advance which parts of the variance we want to retain and which parts we don't. And if we don't measure those, or if we don't know what they are, because we have some kind of we're looking for discovering subtypes or variation across cohorts or something like that, then it's going to be difficult for us to apply it in this context. In our hands, what we've found is combat usually removes too much variance. And so what we do instead, and this is the approach that Mosey developed, is we use a Bayesian model that is, you can think of, it's a hierarchical Bayesian regression model, and you can think of it as finding an optimal balance along a spectrum between not pooling the data at all. So we just take each site independently, and at the other extreme, complete pooling the data. So where we say that we have everybody come from the same site. So we have shared variance parameters. I'm not going to get into the details too much, but basically we this partial parameter sharing between the different sites, which helps us to transfer information from one site to another, basically. And we don't remove any variance from the data at all. So we pass the idea is that we parcellate the total variance into different variance components according to different sites and so on. This also allows us to work in a transfer learning setting, so where we can transfer information from one site to the other. in a way that doesn't require we have data at training time. So we can just transfer parameters from one to the other, and we can use information learned on a large data cohort in a smaller clinical cohort. Mosey evaluated this data on a very large multi-site sample. Excuse me, and this shows you, this is the accuracy, the regression accuracy. So you can see in terms of correlation, this approach at the bottom performs pretty well. In terms of standardized mean squared error, which should be lower is better, it performs pretty well. And similarly for the mean standardized log loss, which measures the variance as well as the mean, it performs pretty well. When we apply this in a transfer learning setting, you can see it performs better than everything else. So it says, okay, we do better than pooling. We didn't apply combat in this context because of the theoretical problem that I pointed out before. And just in terms of, for example, using a prior distribution, we get very poor estimates of the mean. So we get pretty good estimates of the mean, but the variance is really bad. So this histogram is pushed all the way off to the right-hand side here, and it totally removes, at least in a linear setting, all the site effects. So you can't classify the site labels at all afterwards. But it retains clinically relevant information. So this is an anomaly detection approach, which it's not a supervised learning approach. It detects patients as different from the normative model. You can see that it detects all of these disorders, well, apart from ADHD, relatively well. This is the area under the curve for each of these different regions, and it's largely in the pattern of regions that you would expect for each disorder. So the last thing, I think I've got 5 minutes left. The last thing I'd like to talk about is as an approach, some more recent work from Mariam Zabiti. And this is a deep learning based model that aims to find latent representations for brain function. And we use the idea is that we use a convolutional autoencoder and that takes the data, compresses it, and then reconstructs it. And the idea is that in the middle, in a similar way to PCA, we have a representation of the data that captures the essential features and we can use for subsequent analyses. Mariam applied this to the Human Connectome project data. It has a number of different fMRI tasks. We took the cognitive, the fMRI data. The reason that's a good choice is because it provides pretty good coverage of the brain, so that all of the brain networks are covered. It's done so by design, so we can reconstruct different, many different cognitive settings. And this is the architecture. So it's a couple of convolutional layers down sampling. It's not a very deep architecture. There's lots of different validation that she did. I won't talk about that in any detail. What she did then is she took this middle layer, this latent representation, applies manifold learning. It's a little bit similar to TSNE. If you've come across this before, it's slightly superior. This UMAP approach to find representations within this compressed representation of the data. So the first thing we're comparing to PCA. So this is UMAP applied to PCA decomposition, because of course you can think of PCA as just a linear autoencoder. And you can see that this convolutional autoencoder pulls apart different tasks much more cleanly, finds a clearer representation of the different cognitive tasks. Also within tasks, it shows a clearer separation of different tasks as well. That also maps onto behavior. So I'm showing you here associations with cognitive scores. The blue line is the autoencoder and the red line is PCA. And in every case, apart from this task, you see stronger associations with behavior than relative to a linear model. So it seems that this autoencoder is really learning something that's relevant to behavior. We can also do transfer learning in this context, and Mariam did this, applied this to UK Biobank, which has the same fMRI task. And she derived lots of different associations with cognition and lifestyle variables and so on. This is just a Manhattan plot showing the association in a mass univariate setting between this latent representation and all these different cognitive and health-related outcomes. taking this forward, this is really quite a promising way to, for example, to do large-scale normative modeling, anomaly detection, as I've shown you before, and to provide a generative model for brain function across different tasks. So I'd just like to summarize. I'll wrap up there. This is, I won't go through all these points in detail. I've just shown you a flavor for some of the work that we've been doing. I'll just point out as well that we have most of the methods that I've been describing are implemented in a GitHub repository, a toolkit that we've developed. I would recommend people to take a look at. We're well represented here today, so both Sage and Richard will be giving a talk later on. Whoops. So I'd encourage you to sort of visit those as well. And I'd just like to finish by saying that I feel very blessed that we really have a great team. So we have lots of people that have contributed to all the work that I've shown you. It's lots of different skill sets. Everybody works together really well. And I'd like to give full credit really to the people who actually did the work. And with that, I'll stop and thank you for your attention.
Thank you, Andre. That was fantastic. We have actually some time for questions and answers. So please unmute yourself.
Could you explain the logo of the lab, what it's supposed to mean, Andre?
It's a that's a good question. So it's a schematic of a brain that gives a reflection that's analyzed by a statistical model. There you go.
So it's like it's the
It's like a
centile.
shadow. It's a shadow that's described as a statistical model.
It represents you very well.
I had a quick question. Thank you. That was actually really formative, very helpful. I remember seeing your back and forth with the Drysdale and Carl-Liston's group, so that was very interesting to read. So one thing that I had a question about was that combat is in part like identify or fitting these parameters that help regress out batch effects, and while like making sure that if confounds that about that you want to keep, like biological variables, are included in that modeling. Is it possible that you could just like embed it in general in a regression, so that like whenever you do like a regression analysis, if you're looking at whatever new variables, instead of having like something where you like regress, where you're taking out the batch effects, because essentially you're working on residuals after you do combat, but could you just rerun combat every time you're
you could. I mean, the message, but the message, by the way, is not don't use combat. I think combat works well if you're using it for the right purpose.
Right.
The message is that be mindful of what you're actually regressing out. So so the problem that we typically see with combat is that it takes too much variance away, and that one way to think about that is in terms of Venn diagrams and how much correlation is there between age and the things that I'm interested in. And particularly for discovery science, where you don't know you're looking for subtypes, or you're looking for axes of variation that you can't measure, because if you could measure them, you wouldn't do the analysis in the first place.
Right.
So if you do that, then when you do combat, you take away a lot of that variance. It's unavoidable, just by the way that the problem is set up. So that if you keep applying combat repeatedly, you're going to remove more and more and more and more variance. So I wouldn't necessarily, but adding the confounding variables to the subsequent analysis is kind of a little bit what we try to do. Rather than removing variance, we keep it in the model and try to model as much of it as possible.
I see.
Cool. I just have a question, if I could, real quick. That was awesome talk. I actually have two questions. Have you done any normative modeling with fMRI data, or do you have any opinions on what the best metrics would be to do a normative modeling with fMRI? And then the other question is, do you think we should expect to find discrete groups, diagnostic groups, or is a dimensional approach better?
Well, I would probably, so to address the dimensional versus categorical approach, I think it's quite unlikely to find clearly distinct phenotypes. And I suspect the analysis that I showed you using cortical thickness data in autism probably partially reflects partitioning continuous variation. So I think an approach that's agnostic to that is the way forward, and that's one of the approaches, that's one of the motivations behind normative modeling, is that we can model clusters in the data if they're there, but we're not bound a priori to doing that. So the problem with clustering is that clustering always gives you an answer. So you ask it for 4 clusters, it gives you 4 clusters, and then the work that's like the first step, then showing that it's meaningful and etc. is the work that you have to do afterwards. Regarding your first question, yes, we have applied to fMRI data. It's yeah, I think it's a promising way forward. It's of course you have lots of other kinds of structured variation that you need to take into account in that case. Doing good denoising is important, because otherwise you end up with a fancy over-engineered head motion detector, you know, so that kind of stuff is quite important as well, but it has to be taken on a case-by-case basis, really, I think.
Thanks.
Any other questions? So besides the theoretical issues Andre mentioned, I would also like to quickly mention that in my personal opinion, the software implementations may not have been sufficiently validated to rely on. Okay, so what we see here so far, you know, it could be based on probably incorrect implementation. So one of the things, one of the project pitches I have this afternoon is working with, you know, Howie has an implementation. I think Joanne has an implementation too, is to try coordinate an approach to basically validate them as well as integrate them in an open source library I'm trying to develop. I think that without such a common openly validated library, we may be basically talking about different things, although we may be in the same theoretical page, but on a practical data numerical side, we may not be. So keep that in mind. And this applies to many other aspects of our analyses too, but I have more information that I can't publicly say, but I think that we have to be very careful with some tools out there. So if we don't have any other questions, I'll give it over to Tim, who will chair the next session on Meet the Labs. I'm really looking forward to learn from all these exciting labs from Pittsburgh.
Thanks very much for having me.
Andre.
I'm going to have to drop off the call now, but it's been a pleasure, pleasure talking to you today.
Yeah, thank you
Andre.
so much. That has been fantastic.
Thanks, everyone.
Have a good one.
You too. Bye-bye.
All right, so